cut off at a convenient moment. Electromagnetic radiation, also known as light. Stuff in the way of my feet down there. You've probably seen diagrams like this before, where you have long wavelength, short wavelength, high frequency, low frequency, high energy, low energy. That's important. And this tiny narrow band in the middle, red, orange, yellow, green, blue, indigo, violet, good grief, and then ultraviolet on the high energy side of that. Your visible light range, there's some fuzz around the edges, partly because you have red cone cells that see that range, and green cone cells that see that range, and blue cone cells that see that range, and also rod cells that see that range, which means you can see with your black and white only night vision cells, of which there are none in the centre of your eye where you focus on stuff, but plenty around the edges where being able to see things trying to sneak up on you at night is, or at least used to be, extremely important. Wavelengths that are too long or too short for your cone cells to pick up. Does that mean you can see infrared and ultraviolet? As long as you're not looking directly at it. Visible light being part of the electromagnetic radiation spectrum also means you are seeing radiation everywhere you're seeing anything, and infrared light being part of it means you're giving off radiation because you glow. Ever seen a thermal imager picture where people glow? Yeah, that's infrared radiation. You're giving off radiation. It's harmless, don't worry about it. Why this particular wavelength band? Well, it just happens that just so happens that our sky blocks an awful lot of radiation from the sun, but it lets these wavelengths through pretty well. So those are the wavelengths that get to the surface and are therefore potentially useful for looking at stuff on the surface. Useful ones to be sensitive to. Way out this way we have 1 micron. Don't know anything that uses micron radiation. Infrared, maybe. 10 micron, 100 micron, 1 millimeter. You may have heard people talk about millimeter wavelengths slightly. 10 millimeter, somewhere here, around here somewhere, is 60 gigahertz. The velocity is the wavelength times the frequency, and we know the velocity is 299,792,458 meters per second. So 60 gigahertz, if I just put that in, (4.99654 millimeters). So yeah, about there. Been in use since 1992 as proximity radar on cars. You know when you're parking a car and it goes bleep because there's something close behind you? That's what it's been using for nearly 30 years. Your microwave oven, it's about 12 millimeters. Sorry, it isn't, it's about 12 centimeters. I put that in the wrong place. Your microwave oven is way off that way at 12 centimeters. There, like a brain. 12 centimeters, not 12 millimeters. There. Microwave ovens. Once upon a time they were 350 watts. These days you get 800, 900, 1 kilowatt. 1 kilowatt is about how much energy we get from the sun per square meter on average. And of course, that's an average of day and night. And during the night you don't get a lot of energy from the sun. So during the day you get a heck of a lot more, especially if you're in the tropics, you know, pretty much where the sun goes pretty much overhead, and it's near noon, near local southern noon, so the sun is pretty much directly overhead. You get a heck of a lot more than 1 kilowatt, as in big microwave oven, per square meter. And you survive. Because most of that radiation is in these wavelengths. But you might get sunburned because of this. Higher energy. Enough energy to damage DNA. Different atoms, different elements, different molecules will absorb different wavelengths of light. And when heated, give them off. This is why hydrogen burns pink. It's why salty water boiling over from your pan because you took your eye off the pasta for a second and hitting the gas flame goes the exact same shade of orange as old streetlights. Because sodium shines that shade of orange. DNA happens to absorb certain wavelengths of ultraviolet light very strongly, which is potentially problematic because DNA is the recipe book for how to be part of a living thing. And if the recipe for when to stop reproducing because there are plenty of you already gets broken and a cell no longer knows when to stop reproducing, that cell will just keep reproducing and reproducing and you get a thing called a tumour, which with certain slight changes becomes cancer. So yeah, problem. But that's on this side, the high energy side, which can be demonstrated with a gold leaf electrometer or some such marvellous term. We have a flask with a pipe on the side and a rubber bung with a hole through it. Pipe is connected to a vacuum pump which sucks out most of the air, pumps out most of the air, shoves out as much air as it can, allowing the molecules or air molecules in here to bounce off and go that way. Bounce off each other and occasionally bounce down the pipe, and very few of them come back, reducing the air pressure in the flask. Stuck through this hole, you have a bolt. And on the bottom of it, you have two leaves, very thin, very lightweight, very electrically conductive and very tarnish resistant gold foil. And they just hang down. But if you put an electrical charge on this, if you charge something like rubber cloth on a plastic rod, silk cloth on acrylic I think works, one way around or the other. One of those two items will take electrons from the other and pick up what we call a negative charge, because the guy who first discovered this just arbitrarily labelled one positive and one negative and he got it the wrong way around. So it has a surplus of electrons as a negative charge. If you put the negatively charged object on here, the negative charge will be shared with all the metal of the bolt and the gold leaves, and the gold leaves will repel from each other. And that also applies with positive charge, works either way. But there is something that works differently with positive and with negative. If you shine light on it, I mean you've already got light shining it because you're working in a well-illuminated laboratory when you do this, nothing happens. Shine infrared light at it, nothing happens. Shine a kilowatt infrared laser at it. This is a very, very bad idea. You don't want to be in the room where someone is shining a 1 kilowatt laser at anything. It'll reflect off it and it'll probably warm something up somewhere where a lot of that infrared light is bouncing off and hitting, whatever it hits. You might well cause problems with that. If it reflects into your eyes, you are going to blind yourself, but it's not going to affect these gold leaves. Shine a microwave on it. There you. No big deal. Shine 60 gigahertz 5G radiation on it. Nothing happens. Red laser. Yellow light. Orange, you get a sodium light. Sodium lamp. Nothing. Green laser. Blue laser. Nothing. None of these wavelengths will affect it. Get into the ultraviolet, and there is a threshold somewhere here. Above that threshold, that way of that threshold, individual photons have enough energy in them to knock the surplus electrons loose from here. Once you get a wavelength short enough, frequency high enough, and energy per photon high enough to knock those surplus electrons loose, your light can remove the negative charge from this and allow those two leaves to swing back together. But it doesn't happen with visible light or infrared or UVF, or UHF, sorry, or VHF, or HF, or LF, or VLF, or ULF, or microwaves, or radars. It only happens with the very high energy ones. There's another effect which is not related to the energy of the photon, but is related to the wavelength. Imagine going to the beach, or going to a swimming pool at 5 in the morning when there's nobody there and the water's dead calm. More useful. And you stand a steel reinforcing bar in the water, in the swimming pool. Make it a clean one, people get upset if it's dirty. And also a dirty grey scaffolding pole. Or a telegraph pole, if you can get one of those into your local swimming pool. You stand them in the water in the shallow end, you go down to the deep end with a plank of wood, and you create ripples. That wavelength. Arbitrary. Those little ripples come up and they will hit these. If you make tiny ripples, if you're making tiny little ripples like this, they're going to hit this and they're going to bounce right back off it. They're certainly going to bounce off this. On the surface of the pool, you will have incoming ripples, and then ripples with gaps in them that have gone past or between these, and you get that spreading effect because ripples don't just have sudden ends. You get interesting interference patterns behind these objects. And you will have reflected ripples radiating back. Take them to the sea, stand them in the shallow water at the sea on the beach when you have a wavelength like this, and you have one big wave with a trough there and a wave out here is going to wash right past them. You are not going to see a huge great Atlantic roller reflect off a rebar. It's just going to wash past it as if it isn't there. If you get a wavelength that's in between the two, if it's bigger than that but smaller than that, or if it's just, you know, that size, that wave can reflect off your telegraph pole but not off your rebar. Same applies to light when you're looking at stuff. Visible light, that's 500 nanometer green light, can be used to resolve stuff down to about 250 nanometers. Vaguely. It's useful down to maybe 500 nanometers, maybe 1 micron. It won't show you 200 nanometer stuff. Violet light will, but only just. 150 nanometers, you can't see it with visible light. 100 nanometers, 50 nanometers, 25 nanometers, 10 nanometers, visible light is no use at all. What you need is something kind of like a radar or an X-ray machine with really incredibly good resolution working in these wavelengths. Electron microscope. Once you've got one of those, you can start using it to see stuff. Just as you use a thermal imager to view stuff in infrared and detect the infrared and output visible light which we can see, the electron microscope is detecting these wavelengths and outputting visible light that we can see with a heck of a lot of magnification because we're using it to look at very small objects. Very small to the tune of 150 nanometers, 50 nanometers. And you get individual proteins forming an angular case, and then more proteins forming a helix, which extends quite a long way from that case, and then more proteins forming weird spikes, and more proteins forming lumps down there, and inside this you get a little tiny bit of DNA. And the whole thing is too small to see with visible light. But you can see it with an electron microscope, and it'll infect bacteria. That's a bacteriophage design, lambda or mu or whatever the heck. This thing does not move around. It doesn't change shape, except in one very rare instance. It doesn't reproduce on its own. It doesn't respire, it doesn't take in nutrients, it doesn't excrete waste products, it doesn't grow, it doesn't react to its environment except in one very special case. So by that standard, it ain't living. But these proteins here happen to be of a type that can interact with another protein. Another protein that's found on the surface of a bacterial cell. So, surface protein, yeah. Cell membrane is a phospholipid bilayer. And people hear that and go, a what? Right. Beef fat. That's what we typically draw like that. Something like that. What the damn thing's called now? Glycerol molecule with 3 stearic acid side chains, which you may see drawn very much like that. Stearic acid. What the hell is stearic acid? Fair enough. Where can I draw stearic acid? Over here. You may have heard of methane, and you may have seen methane drawn like that. And methanol, not an accurate depiction of the shape of the molecule, but the structural formula, like that. And methanoic acid. Can you have methanoic acid? I think you can. Like that. Very bad for you. Also known as formic acid, as in ant bite. You can also have ethane, which is a lot like methane, but it has 2 carbons. There's a reason chemists don't bother writing in all the H's, you know, we get sick of it. And you can have ethanol, which you won't often see drawn like this, because usually they put the OH on the end, but it doesn't really matter because this is only a structural formula and it's not really that shape anyway. Ethanol is grain alcohol. It's the stuff in beer, cider, wine, whiskey, gin, vodka, whatever. And you can have ethanoic acid, which I am going to draw a different way around from that, which is also known as acetic acid and is found in vinegar. I already mentioned acetic acid. Glacial acetic acid is just a term for very concentrated ethanoic acid. And it's dangerous. Useful, but dangerous. And you can have propane, which also has lots of hydrogen on it. And you can have propan-1-ol, where the OH group is on carbon 1. You can call this 1 and that 3 if you like, but they're the same thing. Or propan-2-ol, where the OH group is on the middle. Propyl alcohol, isopropyl alcohol, hand sanitizer. And you can indeed have propanoic acid. Stearic acid, beef fat, it's a lot like this. Sorry, like this. Except instead of being 1 or 2 or 3 carbons, it's 18. That's beef fat. That's fine. If you knock off this one, just remove that stearic acid and replace it with a phosphate group, PO4 I think. It's been a while. Then you have a monophosphated glycerol bistearate, or some such term. This is a very polar molecule. Don't want to explain polar molecules right now. This has electrons shifted around, right? Your phosphor atom has more protons than electrons. Your oxygen atoms have more electrons than protons. You have electrical charges. Water, likewise. You have an oxygen with 2 inner electrons. It has one of its own electrons and 1 electron from a hydrogen, shared with the hydrogen out there, and the same over there, and 2 of its own electrons there, and 2 of its own electrons there, except it's in 3D. So this one will be up here, and this will be down there somewhere. So you have 8 protons there, 1 proton there and 1 proton there, and 10 electrons here. This side has 10 electrons and 8 protons. This side of the molecule has 2 protons and no electrons. So while the molecule doesn't have a net charge, because it's 10 protons, 10 electrons, the protons are distributed more that way and the electrons are distributed more that way, and it has polarity. Same applies to this. This part of it is polar, and therefore gets along quite well with water. This part is basically this stuff. There's no polarity there. These are balanced, symmetrical. And therefore they don't get along well with water, they get along well with fats, including with each other. So you get these arranged side by side with all your phosphate groups on this side and all your fatty lipid groups on that side, forming a phospholipid monolayer. This kind of works as detergent. You could have a blob of fat in the middle, coated in this stuff, and it becomes soluble because of this double compatibility. In a cell membrane, you have 2 layers of this, with the fat on the inside and the water-compatible groups on both outsides, which was probably a needless diversion. But anyway, proteins embedded in it, this thing bumps into those proteins, and these proteins react to contact with those proteins like an antibody latches onto an infectious agent. Those will be relevant later. Or like an enzyme latches onto a particular part of a protein, or to the end of a protein, or like a signaling molecule latches onto a receptor site. Hormones, they exist. And the response that this whole protein apparatus has to this contact is to fire that DNA down this tube into the bacterium. And inside a bacterium, which may have cilia sticking off it, it may have a flagellum or multiple flagella, it may have all kinds of weird things. It also has a loop of DNA, which has all its chromosomes around it. And it has a mechanism by which it grows. Oh, yeah, important detail. It also has a cell wall, which is kind of like a fishnet body stocking. Stop giggling in the back. It really is kind of like a fishnet body stocking. Made out of pretty strong cables. And as the bacterium grows, this cell wall is also enlarged. It has to be, because the bacterium is full of stuff. Cytoplasm. There are proteins in it, there are fuel molecules in it, there are reserves of amino acids in it, there's all kinds of stuff in there. Outside is water. What happens if you put a large salt crystal or a sugar cube on a damp surface? It gets wet. Deep, difficult questions here. Meaning water is moving from the pure water on the surface into the salt that's, you could think of as an extremely concentrated solution, and there's almost no water there at all, or the sugar. Same applies here. Water, this has more salt in it than this. Salts, whatever. Water will flow in, which causes this to expand. You can demonstrate this with red blood cells, which you can see with a visible light microscope. If you put them in a test tube with salt solution that's the same amount of salt, the same concentration of salt as what's inside the red blood cells, they don't change. If you put them in a very concentrated salt solution, water will flow out and the red blood cells shrivel up. If you put them in nearly pure water, water flows in and they swell up. Put them in pure enough water, water flows in and they swell up and burst. So the bacterium has to have this cell wall around it to stop it bursting. Some also have an outer membrane. If you ever hear of gram-negative and gram-positive, that is whether the gram chemical will stain the bacterium. Gram is a stain that binds to the cell wall. If there is an outer membrane in the way, it can't, so they don't stain, so they're gram-negative. If there isn't, it can, they do stain, it's gram-positive. Yeah. Penicillin, you've probably heard of. Penicillin prevents the bacterium from making more cell wall. As the bacterium grows, eventually, and can't produce more cell wall, eventually the gap in the cell wall gets so big that the bacterium can burst through it and die. Which obviously means penicillin isn't going to work on every bacterium, because it can't get it here, and it really isn't going to work on a bacterium that has an extra loop of DNA, extra chromosome, in fact, with a gene on it, for an enzyme that destroys penicillin. If the bacterium finds penicillin and goes, om nom nom, I'll have those resources, rather than dying, penicillin ain't going to kill those bacteria. What the virus does is inject its own little chromosome, which contains genes that reprogram the bacterium, they're coded to be always on, high priority, to turn all its available resources into lots and lots of viruses. So it produces lots and lots and lots of viruses, and it encases each one in its own little protein shell. And there are a couple of things that can happen with some viruses in humans. What it will do is actually push the viruses out in pockets of envelope, and you get enveloped viruses, the membrane around the outside of the protein shell. With bacterial viruses, bacteriophages, what tends to happen is it produces a couple of hundred of these and uses up so much resources that it falls apart. That bacterium cell has been killed, but there are now hundreds of copies of the virus that infected it. Without the virus ever being alive, it has reprogrammed the bacterium to create lots more of the same virus. It gets really exciting when there's a mistake and this chromosome break, and they merge together and then they break differently, and the viruses that come out are actually carrying a gene from here. Like the gene from cholera that encodes the poison that makes it give you the shits. You have a virus going around that can now infect other bacteria and inject them with that gene. Why does it matter if it blows up the bacteria it infects? Well, your agar gel plate with your little clear patches where the virus has killed all the bacteria, sometimes you find little tiny white specks of bacteria it hasn't killed, where it's gone in and then it's gone, nah, I'll just sit here for a few million generations. And you get 2 to the power of however many hundred generations it is, copies of the bacterium that all have the viral genome as well, including that stolen gene. Could be significant.